They say there is a product for it now, a capsule, a powder, a compound with a sleek label and a promise large enough to sound biological. Activate autophagy, clean out damaged cells, and rejuvenate the body from the inside. It is one of the most seductive ideas in modern health culture. Because autophagy is real, it is one of the body's most elegant cellular systems, a recycling mechanism in which worn proteins, damaged organelles, and molecular debris are enclosed, transported, and broken down for reuse. It is so important that Yoshinori Ohsumi received the 2016 Nobel Prize for uncovering its mechanisms. But the existence of autophagy and the claim that one consumer product can simply switch it on are not the same thing. Source. So what does science actually show? Is there a best product? Or is the real story happening much deeper? Inside nutrient sensors, lysosomes, and the cell's own survival machinery. The first quiet decline. They are crowded workspaces. Proteins are folded, unfolded, damaged, repaired. Mitochondria produce energy, but they also generate oxidative byproducts. Membranes wear. Molecular fragments accumulate. Every day, inside every tissue, tiny forms of damage appear simply because life is active. This is normal. The problem begins when cleanup falls behind. When damaged proteins are not cleared efficiently, they begin to collect like dust in a machine. When faulty mitochondria remain inside the cell, energy production becomes less efficient and oxidative stress can increase. The cell does not collapse immediately. It becomes noisier, less precise, more burdened by internal friction. When these are the first silent changes, not dramatic symptoms, not sudden illness, just a gradual accumulation of biological clutter. This is where autophagy matters. Autophagy, literally self-eating, is the cellular system that encloses worn-out components in membrane sacs and sends them to lysosomes, where they are broken down and recycled. It is not a trend. It is not a hack. It is part of the body's baseline housekeeping machinery. Source. So when a product claims it can activate autophagy, it is speaking to something real. But real biology is rarely as simple as marketing. The molecular lock's autophagy is governed by molecular locks. The most important of these is mTOR1, a nutrient-sensing complex that tells the cell whether conditions are abundant enough for growth. When amino acids, glucose, and energy are plentiful, mTOR1 remains active. And when mTOR1 is active, autophagy is restrained. Growth takes priority over recycling. The counter signal is AMPK, the cell's energy alarm. When energy availability drops and the AMP to ATP ratio rises, AMPK activates. It suppresses mTORC1 and promotes proteins such as ULK1 and BCLIN1 that initiate the autophagy pathway. In simple terms, the cell shifts from building mode to maintenance mode. Source. This is why the search for a best product is biologically misleading. Autophagy is not a button sitting on the cell surface, waiting for a supplement to press it. It is a system's response to nutrient status, energy sensing, and internal stress. A compound may influence one part of this network, but the network itself is larger than any bottle. The cell asks a deeper question before it intensifies autophagy. Is energy scarce enough? Is growth worth pausing? Is maintenance now the higher priority? Only then do the internal workers begin the heavier cleanup. How the recycling system works. Once the signal is given, the cell begins construction. A curved membrane starts to form around damaged cellular material. This early structure expands and gradually encloses misfolded proteins, defective mitochondria, and other worn components. When sealed, it becomes an autophagosome, a double membrane transport vessel carrying biological scrap. It is the body salvaging value from damage. The Nobel Prize press release described autophagy as the process by which the cell encloses its own contents in membranes and sends them to the lysosome for degradation. While Ohsumi's work showed that this machinery is ancient, conserved, and fundamental to human health. Source. So when people speak of rejuvenating cells, the scientifically accurate picture is not one of magic renewal. Chapter 4. The silent deepening autophagy does not usually surge under conditions of abundance. It deepens when the cell senses scarcity. Fasting and calorie restriction are among the best studied triggers because they reduce glucose and amino acid availability. Lower ATP, raise the AMP to ATP ratio, activate AMPK, and suppress mTORC1. That biochemical shift opens the gate to greater autophagic activity. Source. But even here, the process is not binary. Autophagy does not suddenly appear from nothing. It exists at baseline, then intensifies as conditions change. And like many maintenance systems in biology, its benefits depend on degree and context. The same review literature that describes fasting-induced autophagy as adaptive and protective also warns that excessive or prolonged autophagic responses can become harmful, contributing to cell death pathways when the degradative load exceeds what the lysosomal system can safely handle. Source. This matters because product marketing often treats autophagy as if more is always better. But the body does not think in slogans. The body thinks in balance. Too little cleanup allows damage to accumulate. Too much degradation under the wrong conditions can also injure tissue. The biology is not about maximizing one pathway forever. It is about regulated maintenance, quiet, timed, proportional. The product myth. This is where the science becomes uncomfortable for marketing. There are compounds that influence pathways related to autophagy. Rapamycin is the most famous. It inhibits mTOR, and in laboratory and animal research, it is deeply important to aging biology. Human studies have shown some promising effects in immune, cardiovascular, and skin-related measures. But the evidence is mixed, system-specific, and far from a universal rejuvenation story. Human trials also report trade-offs, including more infections in some populations and increases in cholesterol and triglycerides in people with age-related disease. Source. Then there is spermidine, often sold as an autophagy supplement. Preclinical work is intriguing, but human evidence remains limited. One human study found that oral spermidine significantly increased plasma spermine rather than spermidine itself. Honorine and the authors explicitly noted that short-term effects from typical supplement doses below 15 milligrams per day are unlikely. The same paper cautioned that clinical data have not confirmed the anti-aging benefits suggested by preclinical models. Source. So what is the best product? Scientifically, the answer is unsatisfying but clear. No over-the-counter product has robust human evidence proving that it reliably activates autophagy and rejuvenates cells in the sweeping way advertisements imply. What long-term biology actually cares about autophagy matters because cells age. Not emotionally, mechanically. Over time, protein aggregates accumulate. Mitochondria lose efficiency. Cellular signaling grows less precise. Aging can be understood in part as the gradual wear of maintenance systems. Autophagy is one of those systems. When it functions well, it helps preserve cellular order. It removes defective components before they become larger problems. It supports tissue resilience. It is implicated in how the body manages inflammation, metabolic stress, and age-related cellular decline. The review literature consistently describes adaptive autophagy as protective housekeeping. Source but long-term biology does not revolve around one lever. The cell is an ecosystem. Nutrient sensing, protein synthesis, mitochondrial turnover, lysosomal function, inflammation, insulin signaling, and circadian timing all interact. Autophagy is one essential worker in a much larger system. That is why the idea of a single best product is so tempting and so reductionist. The body is not a lock waiting for one key. It is a city of interdependent processes, each responding to the others, each adjusting in real time, each balancing growth with repair. Rejuvenation, if the word is used carefully, is not a single event. It is accumulated maintenance, reading the body's signals. The body rarely announces autophagy directly. There is no sensation that proves it has intensified, no sudden internal chime, no unmistakable sign that the recycling crews have started their shift. What people often feel instead are the conditions surrounding it. During fasting, a wave of hunger may reflect ghrelin rhythms. A lightheaded period may reflect fuel transition. A sense of post-fast clarity may coincide with changes in glucose handling and rising ketone availability. With digestive heaviness after certain supplements may have nothing to do with cellular renewal and everything to do with gastrointestinal response. These are not proofs of rejuvenation. They are metabolic signals. And that may be the most important correction of all. The body does not sell spectacle. It runs maintenance in silence. Autophagy is real. It is one of the cell's most elegant survival systems, a recycling process that helps clear damaged material and preserve internal order. But the idea that one product can simply switch it on and rejuvenate the body is not supported by strong human evidence. Rapamycin remains a serious drug under study with trade-offs. Spermidine remains promising but unproven at the level marketing often suggests. Source. The body gives signals before breakdown, and the science gives signals before certainty. Understanding the body is the first step to respecting it. Health is not sudden. It is cumulative.